right, looks like it's 3:05, so we can begin. Hi everyone, my name's Krish. Thanks for coming to our event. Before we begin, we'd like to introduce our speaker, Dr. Christopher Bahl, share some words of gratitude, and explain how this event will work. Dr. Christopher Bahl is the head scientist of the Institute of Protein Innovation and a lecturer at Harvard Medical School. His work focuses on utilizing modern computer science, like computational design methods, supercomputing, and even robotics, to develop proteins which can neutralize infectious diseases, recycle plastics, and even potentially cure cancer. He's also a TED Fellow, one of the 500 remarkable thought leaders globally recognized for his exceptional accomplishments and innovative breakthroughs. Thank you so much for coming today, Dr. Bahl. We'd also like to thank our club advisor, Mrs. Martin, for her invaluable guidance and support, as well as our ISS event team, Sanjay, Ray, Aditya, Anuj, Anas, Rishab, Srinidhi, Preet, and Naman, for putting in the hard work to make this event a reality. The way this event will work is that we'll begin with a 20 to 30 minute presentation from Dr. Bahl on his research and career tips, and then we'll use the remaining time for Q&A.
Great. Everybody can see my screen okay? No. Great. Okay. So the title of my talk is fairly simple. Protein design is amazing. I really, I feel like I don't actually have a job that I trick a bunch of people into paying me to do my hobby every day. And yeah, hopefully you'll convince at the end of this talk that protein design is amazing as well. So you may not be familiar with what protein design is, and like most people, when they think of protein, they think of food. But protein is so much more. So let's talk a little bit about what you'll learn today. So the first thing is we'll cover proteins, and I'll describe their molecular structure. Then I want to try to give you a framework on how to think about proteins and convince you that proteins are nano robots. Then we'll talk about how to design these nano robots and some of the challenges. And then finally, I'll just sort of tease some of the cool stuff that we're working on designing these nano robots to do. And I'll wrap up and give a little bit of advice on folks who are interested in going into science in the future. Okay, so just to, hopefully this is review for lots of folks, but a protein is a string of amino acids, right? So each of our cells produce proteins, and they're super important. Proteins mediate nearly all the chemistry of life. When you think about proteins, you think about muscle often, right? Well, muscle is full of protein that does all the stuff that moves your body around, right? So if you look at an individual protein, one molecule of protein, it actually looks a lot like bead on a string. There are 20 different amino acids that your cells can use to build proteins. And you can think about breaking these down into two different types. There are some amino acids that repel water, they're oily. The scientific term here is hydrophobic. And there are some amino acids that like to interact with water, and they sort of attract well and behave well with water. So these are hydrophilic, so we call these things they like water. And so now if you take this string of protein and you stick it in water, instead of being a long, unstructured, just kind of happy string, a good protein will adopt a specific shape. It's still one continuous string, but it's no longer just kind of flopping around randomly. Now the amino acids that don't like interacting with water, the oily hydrophobic amino acids, get pushed to the interior of the protein. And the amino acids that like to interact with water get pushed to the outside of the protein where they can interact with water. And so it's this interaction with water is what drives the protein to adopt a particular shape. And here I'm showing it simplified in 2D, but in reality, it's a really intricate three-dimensional structure that this molecule forms. And it's this structure that gives it its function. The proteins are what carry oxygen through our blood. They allow us to digest food. They are in our brain. They're how neurons talk to each other. And in fact, your DNA, the primary function of your DNA is to be an archive of blueprints for building proteins. So super important. Okay, so now let's think about proteins as nano robots. This is maybe a bit of a leap. So let's start early. What is a robot? Probably most people are thinking something like this, right? A very capable robot that's able to do lots of different tasks, complex information gathering and output, really versatile, right? This is probably the first thing. But there's a much more humble robot that many of us probably have in our homes but take for granted, and that's the dishwasher. The dishwasher is definitely a robot. You put in your dishes and out come clean dishes. But we don't, I think most people would never think, oh yeah, I have a robot in my house, right? And think about a dishwasher. So a protein is more like this kind of robot, a dishwasher, rather than an automaton kind of robot. But the exciting part is when we start to put many robots together that are more like a dishwasher. And so your cell, you can think about, is a collection of many different, more simple robots. So here's a picture of the Tesla factory. And you can see there's lots of robots that all work together in order to build remarkable things. And so this is more like the true nature of how biology operates. And when we think about designing proteins and designing things that do stuff, we're really thinking about building one small robot in this large factory. And how can that change the properties of what the factory can do? How do we string multiple robots together to create new, larger pieces of function? Okay, so now let's dive into this analogy a little bit further. So our dishwasher is a robot with certain properties. First, it needs a blueprint for assembly. The shape and the structure of this dishwasher dictate the function, right? It's a box. The box is for keeping the water inside so that it doesn't go everywhere. The dishes get loaded into these racks. These racks have specific shapes that hold the dishes. You've got water jets that put water on the dishes, and the angle that those shoot water at the dishes also controls how good it is at washing. It takes input and it delivers output, right? So the input would be dirty dishes, soap, and water, and the output will be clean dishes and dirty water. Input's also electricity. Another important feature is it's programmable and controllable, right? There are multiple settings on your dishwasher. You can decide what type of program you want it to run, and you can control when it operates. It doesn't run all the time uncontrollably, right? You hit go and tell it to start. Proteins are the same. So this here is ATP. So in your cell, there's a really important molecule called adenosine triphosphate, or ATP. This is what you can think of as the energy currency inside of your cells. If a chemical reaction needs some energy in order to be able to go, cells can grab the enzymes in your cell can grab ATP and use this as sort of energy to do work inside of your cell. But you have to make this energy currency in order for anything to happen. And there's this incredibly sophisticated and amazing molecule called ATP Synthase that does it. And this thing is literally a molecular motor. So you've got an axle that spins around inside of this component here. You've got a stator, which applies torque and converts it into chemical energy. And you have a difference in the number of protons on either side of a barrier here, a membrane. And there's a chemical gradient which you use, which this robot converts into and stores it as chemical energy. It's really, really fascinating. So you need a blueprint to build this thing. your DNA serves as the blueprint. Each component of this is recorded in a different gene stored in the DNA of an organism. The shape and the structure of this remarkable molecule dictate its function. So you need to have these components that can rotate. You need to have this stator arm on the side that can hold in place and harness the torque. It takes input. You need a proton gradient, and you need the components in order to build ATP, and it delivers output. It makes ATP. It's also programmable and controllable. So the cells have the ability to switch this molecule on and off. It can run it in either direction, and it can do different things with it. So now for the exciting part. How do you build a protein nanobot? How do we make this do something fun and interesting? So in order to create the function, you have to build the structure. Without these structures, these molecules to operate and do anything. So now let's talk about designing these and challenges to face when we want to create these nano robots. So in order to build function, you need to build structure. So this now begs the next question: how do you build protein structure? And the answer there is to design the amino acid sequence, right? So if we go back a few slides, jump here. We have our chain of amino acids here, right? And you can see that they're not just one long stretch of the blue amino acids that like water and one long stretch of the ones that repel it. You have to have a specific pattern of these different amino acids. And this is simplified with only 2 of them, but there are really 20, so the problem is really complicated. But it's this specific pattern of different amino acids is what allows the protein to adopt that shape, right? So you need to be able to know the order in which these amino acids need to appear on string. And that's amino acid sequence design. And that's the bulk of the problem that I work on every day. So let's think about why this is challenging. So I mentioned a couple times, there's 20 different amino acids. There are some exceptions, but 20 is the thing that pretty much everything that's alive on the planet uses 20 different amino acids to build all their proteins. If you look at the average protein in anything that's alive, it's 336 amino acids long. So that's how many of these building blocks are in this chain, on this beads on a string. You have 336 of these beads on the string. So now if you think about how many different proteins could you make? It's a lot. It's a really big number. If you only have an average protein that's 336 amino acids long, that average protein could have 20 to the 336 power different sequences. This is a number that's so big the human mind cannot wrap around it. It will melt your brain. Let's explore just a little bit about how brain melting this number truly is. The observable universe, physicists have made best estimates of how many amino acids are contained in everything that exists, all of the observable universe. It's only about 10 to the 80th atoms. Our universe is also about 13.7 billion years old. And if you convert that to seconds, now we're 4.3 times 10 to the 17 seconds. The fastest that humanity has ever got a computer to run was about 8.8 gigahertz. And as you can see, it required some rather elaborate cooling measures, so they had to pour liquid nitrogen directly onto the processor to keep it from bursting into flames. But that's the fastest we've ever gotten a computer to operate. So now, let's do a thought experiment. Let's say that every single atom in the entire universe stores a bit of information. So we've got about 10 to the 80th bits. The whole universe is a computer. And that computer is operating as fast as any computer has ever operated. So about 8.8 gigahertz. And that computer has been operating continuously since the beginning of time itself. That computer would have only sampled 3.8 times 10 to the 108 bits. An average protein could have 20 to the 336 different sequences. This means that the entirety of existence, for as long as existence has existed, is less than a rounding error in the potential for the average protein. So what this means is even though evolution has been, even though life has been on the Earth for several billion years, evolution has only sampled a tiny fraction of what's possible to make using protein. Really, to say that it's just the tip of the iceberg is a gross underestimation. Protein can do many fantastic things, and we have really just yet to see what makes it so amazing. Another limitation of proteins in nature is that they're all related to each other, right? Organisms evolve by mutating their DNA, which changes their proteins, and each organism that's alive today is related ultimately to the very first thing that was alive on the planet. We've changed a lot since the very first cells, but we're all connected, we're all related, which means that each protein changed a little bit at a time from the one that came before it. What this also means is if you think about this vast sequence space, this vast potential for protein, the proteins that have existed are not randomly distributed throughout this space. They exist in these small sequence islands, is what we call, where proteins are related to one another. Some people in the lab have made lots and lots of mutations to sort of speed up the process of evolution and been able to push back the bounds and explore regions of protein sequence space and function that nature could never do. But I think that the challenges that we face together as a species are great. And the solutions to those challenges, I don't think all those solutions lie right on the edge near something that has existed before. We're going to need to dive deep into these unexplored regions of potential to find things that are completely different from anything else that has ever existed. And in doing so, unlock new and exciting functions that nature could never create. So let's talk a little bit about protein design, where it comes from. You may have heard about DeepMind in the news recently as an AI company, and they recently had a major breakthrough in predicting the structure of proteins. When we think about protein structure prediction, what we're doing is taking the order of amino acids, these beads on a string, and we're using this information to predict what the 3D molecular structure will look like. And at the end of the day, it's this molecular structure that we care about. That's how we judge accuracy. It's very exciting to be able to understand nature and how natural proteins work. Protein design takes this problem and flips it on its head. We're designing proteins that do some function that we create, that we dream up. And then we find out, using algorithms, what amino acids to put at each position on this string, what identity each bead needs to be, and in which order they need to be placed in order to create a molecule that has this structure. So ultimately we get this protein sequence, which we can then convert back into DNA and then genetically engineer an organism that will then go and produce our protein, which we can test. One of the exciting things about protein design is that it can help us to understand how to build protein structure. So we can take bits of pieces of proteins assemble them together, and then find out and really learn what it takes to do this. And the fun thing is we really exist at the intersection of two new technologies. New, I should say, relatively new. First is supercomputers. So here's a postdoctoral fellow who works in my group, Dr. Frank Tietz. And this is a small fraction of the supercomputer that we built here at the Institute for Protein Innovation. And you can see Frank is very happy with running two computers here at the same time. But really there's many computers more. And it takes a lot of computational power to be able to do these calculations. Also accelerating this is the use of automated liquid handling robots in the lab. And so here is our Hamilton robot. And here are these little spikes. Each of these is hold tiny amounts of liquid that we can mix together. We can use to process samples in the lab. And it's got arms for moving stuff around on the deck. And we can use this for doing genetic engineering, for extracting our designed proteins from cells that we've genetically engineered, and for testing our proteins to see if they do what we've designed them to do. Okay, so that's a little bit about the why and the how, but now let's switch gears and talk about what can we do with all this. It's pretty neat. So one of the first things, and maybe most obvious things to do, is to create medicine. And not only to create medicine, but to understand how to create medicine. So this is a molecule that I think everyone is familiar with. It's an antibody. So if you were to receive a vaccine, really what you're doing is you're teaching your immune system in your body how to make an antibody. This antibody would say, for instance, bind to a virus and make you immune to it. And so there are binding sites on this antibody here and here that it can use to interact with a virus and neutralize it. We can do this by injecting stuff into animals and then harvesting their antibodies and figuring out how the body naturally did it. But the exciting stuff is coming where we're starting to design these rationally. And in doing so, we can be much more precise in how they operate and be much faster in being able to create antibodies with any capability that you might need. We're also designing other proteins that can neutralize the SARS-CoV-2 virus, for instance, the agent that causes COVID-19, and molecules that are durable enough that we can put in the air. So we actually have a collaboration with a nanoparticle engineering lab here at Harvard, who creates structured water droplets that are very tiny and have lots of charge on them, and so they stay in the air for a really long time. And we're loading these structured water droplets in with proteins that we designed to neutralize the virus, with the idea of creating a nano mist or a nano fog that would hang around in the air for a long time, would be really thin so you wouldn't notice that this fog even exists, and it would protect you by neutralizing any viral particles that are floating around in the air. And another project that we're working on is creating proteins that can break down plastic. Plastic recycling is really important. We've really put a lot of plastic into the environment, and we need better ways of fixing this and breaking it down. Let's see, message from Krish, clarifying questions. Let's see, are there any questions that people want to have answered at the moment? No, okay. Okay, so let's that's a little bit, just a taste of the types of things that we're working on creating using protein engineering. But really right now, I think the potential truly for protein engineering is human imagination. What can we dream up? Because it's possible to design nearly anything that you can dream up. Some things to design may be harder than others, but almost anything is possible, which is really exciting. So let's talk a little bit about the future, and if you have a career in science, some general advice. This is a mantra that I hear all the time in science, is that we stand on the shoulder of giants, right? We think about Einstein and von Leeuwenhoek and Pasteur. I think this is wrong. We don't stand on the shoulders of giants. We stand on the shoulders of regular people. There are many scientists that have created many important discoveries, have contributed many important discoveries, have created many important technologies. They're regular people. Anybody can do this. It just takes someone who's excited and passionate, and you can totally do it. So this putting people up on a pedestal, I think is not always a great thing to do. They're certainly great, but they are regular people.
Dr. Bahl, it seems we have a clarifying question in the Mentimeter. Someone's wondering, if new proteins are created through generations, how can we hope to map everything? More and more proteins will keep getting created, right?
Right, yep. So the question is, are we mapping everything? Is that the...
I think the question is asking, how can we hope to map every protein?
So it's not possible to map every protein. There are simply not enough atoms in the universe to map every protein. You would need a universe of universes to even start to approach something like that. So I think maybe the punchline there is that the potential for protein is mathematically limitless. The number is so large, it's effectively infinity for all practical purposes. There's not enough universe for it to be finite. Not sure if that answers the question. Okay. So let's see. Yeah, science is supposed to be fun. There are lots of careers that you could pursue, and there are many easier ways to just earn a bunch of money, have a work-life balance. Science is something I do because I'm a huge geek and I love it, and I can't imagine doing anything else. And so if it hooks you and you get that, go for it. If science isn't the thing that really hooks you, don't feel bad about doing something else. There are many good reasons to do stuff. Find the thing that's you're passionate about, and you'll certainly be happy when you do it. If you do pursue science, at some point you'll join a lab and learn how to do science in that lab. The principal investigator could be a professor if it's at a university, could be a scientist if it's at a biotech company. Look for someone who's happy and enthusiastic and excited to be there, excited about the science that they're doing. And in that lab, you'll have scientific colleagues. You also want them to be happy, enthusiastic, and excited about the science that they're doing. That's a great environment, and that is an indicator that it's a place that you want to be. You'll work on some project as an undergrad. If you stay in science, you probably will go to grad school. You'll work on some other project in grad school. In both cases, very few people actually continue working on that same project after graduation. So find science that's exciting about it, but don't feel the pressure of having to pick the thing you're going to work on for the rest of your life, because very few people will actually do that. Focus more on learning how to understand the systems, the techniques, the skills, and how to think like a scientist. Likewise, training in the current state of the art is good. learning the best, newest technologies. But also, don't be afraid to take risks on technologies that aren't fully mature yet. Something that hasn't quite made an impact, but it's a great idea that someday this new technology will be really important. Because if you're one of the people who makes it important, you can establish yourself as a leader. And finally, mentors are incredibly important at all stages of your career. So this is my graduate as Dean Madden, and this is us having a duel with cryo equipment in the lab. So these are tongs that you use for handling and manipulating protein crystals and liquid nitrogen. Just really taught me the passion and joy for doing science, and really how to think like a scientist. During my postdoctoral studies with David Baker, I really learned how to take a super challenging problem and break it down, and so how to have fun outside of lab through mountaineering. And now at the Institute for Protein Innovation, Tim Springer is really teaching me how to think about making your science as impactful as possible, bringing it to the world, and helping to improve people's lives. And so, with that, I would love to take questions and discuss. And thanks everybody for coming out today and for your attention.
Yeah, thank you so much for your presentation. It was very enlightening to learn about, how proteins operate, how you guys designed them, as well as advice you had for students who were interested in pursuing a similar path. We actually have one more clarifying question with 6 upvotes asking if do new proteins need to be created completely from scratch, or can existing ones be mutated or manipulated to have desirable properties?
Yeah, that's an excellent question. The truth is, it's both. It's not either or. Yep. So protein design really started with engineering proteins that already exist and changing them. And then there's the relatively new field called de novo design, which sort of means from the beginning in Latin. And that's building proteins totally from scratch that have no relation to anything in nature. And so we do both. Yeah, it's a great question.
And also, right before we segue to the Q&A, someone was also asking, what are some examples of problems you've solved with protein design today?
Yeah. So let me stop sharing here. So there's several problems that we've solved, and many problems that others have solved. So with de novo protein design, myself and colleagues, we've designed proteins that can bind and neutralize influenza virus. So that was pretty exciting. We've designed proteins that can neutralize botulinum neurotoxin, Botox. Some colleagues of mine who I work closely with designed some proteins that can stop cancer cells from growing. Many medicines approved by the FDA are proteins that were engineered and changed. Another great example of an engineered, designed protein, there's a new protein that's in clinical trials right now that might cure celiac disease. It's a protein that breaks down gluten, and it's a pill that you can just take, and the protein will work inside your stomach and break gluten down. It's really lots of cool stuff.
Okay, so now we'll be moving to the Q&A segment of this presentation. And to make sure that clarifying questions and Q&A questions aren't being muddied up, we'll be sending out a separate Mentimeter.
Yeah, so like Krish said, I sent up the link for the Mentimeter for the Q&A section. They're different links, so make sure to click on that one if you have questions. The Q&A. We'll be rotating on questions that we see pop up and should have a minimum of 5 upvotes. But if we don't have any questions yet, we'll probably start with a few ones that we have.
So I guess a really general one we've gotten a bit before the event is, what made you, want to be interested in taking on computational protein design?
Yeah. That's a great question. So for me, my interest in protein engineering in particular started when I was in high school taking AP biology. And I was learning about how cancer cells work, and found out that inside of a cell, the environment changes when a cell becomes cancerous. And there are changes that you could detect, in theory, using a protein. And maybe it would be possible to detect those changes and then kill the cell from the inside. And when I was in high school, I thought, oh, well, this will be super easy. All I have to do is just make a protein that does this, and bam, cancer's done. And then I started learning about how hard that would be, and that sort of led me to where I am today, because it's really hard, and because it's really challenging, it's been really fun. And to be able to do something that crazy, there's no protein in nature that is able to do anything even remotely close to that we could repurpose. So we really need to build something new. And the only way to do that is through computer modeling and computational protein design. And so that got me really excited about designing proteins in the computer. And our technologies have finally advanced that to the point where we're just getting started working on this problem directly. Which has been really fun. It's something of a lifelong dream now that's starting to come true. But we still have, I think, many more years of working on it before it will, be successful.
And it seems by the Q&A questions, a lot of people are also interested in using protein design to cure cancer. They were asking, how can protein design help enhance or create new cancer therapies? And what would you recommend as in courses for someone who wants to go into cancer research?
Yeah. So that's also a great question. So there's a couple big ways I would say that protein design can help with cancer. So the first is that your immune system already is really good at killing cancer. And your immune system is really based on proteins. So you have immune cells that have proteins on the surface, and they use those proteins to communicate with cells in the body. And if you can learn to speak cell, the language of cells, then you might be able to talk to them and change how they interact with different cells, right? And so cancer cells tell your immune cells that, hey, I'm cool, don't mess with me. And if a cancer is growing, there's a good chance that it's learned how to do that. And the immune cells are no longer killing the cancer because of that communication. The cancer cells have lied to the immune cells, and the immune cells believe it. You can design proteins that can shout over that lie and cause the immune cells to become active again and start wiping out the cancer. So that's one of the ways that many people are doing it. It's called immunotherapy, and there are several ways. Some of them is putting a molecule in there that will interfere with that communication directly in the person. Another way is to pull immune cells out of a person, genetically engineer them to change the proteins that they have and how they work, and then put those cells back into a person to attack their cancer. So that's one way that involves enhancing the body's natural immune system to fight cancer. Another way is by finding specifically the cancer cells in the body and delivering poison only to those cells and not delivering poison to healthy regular cells. And so there are also lots of ways to do that, many different types of poisons that you can do, but it's a way that you can bring really toxic, bad stuff that you don't want everywhere in your body only to the cancer cells.
And it seems like a couple of people are also wondering, not just with cancer, but how could this process work for neutralizing viruses? And how does the study of protein design help us in isolating viruses like the current coronavirus?
Yeah. So more really great questions. So neutralizing pathogens in some ways is a little bit easier because if you think about cancer, maybe an analogy is like a like an old clock that has lots of gears and springs and it's super complicated. If the clock is a little broken, you need to be really precise in how you change it and fix it, because the clock needs to run again. In this analogy, you are the clock, right? With an infectious disease, for the most part, the infectious organism is the clock in this case. And so the most effective way to solve that problem is to take a hammer to the clock, right? And that doesn't require much sophistication. You just bash it with a hammer. And so that's basically how your antibodies work, by neutralizing viruses. Sometimes if you hit a clock in one spot versus another, it might do more damage if you hit it right in the springs versus if you hit it maybe at like the leg, right? And so sometimes your immune system is not always very good at picking the best spot to hit the clock. But with protein design, we can do that. We can say exactly, find the Achilles heel, the most vulnerable part, and hit it there, and hit it hard.
Got it. And then some people are also wondering how well they would need to know computer science to work on computational protein design.
Yeah. So the fun thing is that maybe 100 years ago, it was possible for one human to know almost all science, like all of it. Like everything that humanity knew about science, one person could pretty much know it about 100 years ago, give or take. Today, we have so much knowledge that in order to do something really cool and exciting, no one person can do it by themselves. It really takes a team of people, each with a different skill set and expertise, in order to do this. So the hard challenge is you need someone who's a computer science expert. You need someone who is a immunology expert, who knows how human bodies work. You need someone who is a protein biochemist, a protein expert who knows about protein structure. And it's this team together that comes and solves the really big challenges. So if you're excited about biology, cool, pursue that. You could definitely work on a team that does these types of things. If computer science is for you, awesome. Like there's a need for that on the team. The more you can understand what your other team members know, the better you can be their teammate. But you don't necessarily have to know how to do all of it yourself. Just being able to kind of speak the language of someone else's expertise is often enough to collaborate with them.
So would you recommend that, for any students who are interested in going down a similar path as you, they specialize on, something they're really passionate about versus try to be a jack of all trades and different things?
Yeah. The truth is it takes both. So if you're doing the thing that you're passionate about, then there's a good chance it'll work out. If you're passionate about knowing some stuff about many things, that's very useful. If you're really passionate about one super specific thing, then that can also be really useful. It takes all kinds.
Got it. I think that's definitely really, key advice for some of the people here, because I think, at least from what I know, a lot of people just try to, get if they're interested in something, they not only try to learn that, but also other related things, the hope that might be useful as well. But really specializing and staying focused on what you're passionate about is really interesting advice. A couple of people were also wondering, what does that average workday look for you?
Oh, an average workday? So I live in Boston, so I commute on the T, come to the Longwood Medical area. There are 4 major hospitals here, research hospitals, so it's a really exciting place with lots of clinicians right next door. And right now I lead a team of scientists that have really diverse backgrounds and really diverse skill sets. And I get to meet with each of them and help them think about the problems that they face in the lab, and brainstorm and talk about science, help them troubleshoot what they're working on, help them brainstorm creative ways to achieve their goals. And then I spend a lot of time also talking to other scientists and other research groups in the area, clinicians and basic researchers and other people who have challenges that need protein engineering, essentially. Many clinicians have challenges of, we could cure this disease if only we had a protein that could do this, function X, whatever it is. And they don't have the expertise to engineer a protein, but they have all this disease expertise. So forming a team to be able to tackle that sort of thing. So I guess that's a long way of saying that my average day is spent in meetings.
How would you suggest that people try to develop those team skills?
Sorry, develop which skills?
Those, teamwork skills that you were talking about.
Oh yeah. If your high school has like an iGEM team, that's a great way to get involved in science and teamwork. Many undergraduate, for those who are ready to move on to college soon, most universities will have iGEM teams as well. iGEM is really great. It stands for International Genetically Engineered Machine. It's a huge synthetic biology competition. It's global, so it's a really great program to get involved in. If you have a robotics club, that's another great thing. Those are like really team-oriented, problem-solving type things. I'm sure there are other types of clubs. If not, then you could follow Krish's example here and form one.
Got it. Yeah, it's definitely interesting. I think when a lot of people think about getting into science, they focus more on learning the subject and understanding the concepts. But as I sort of learned from listening to your talk, it's also a lot about being able to work with others and other specialists.
Yep. For me, I never really connected with the material that I learned in school until I knew what it was for. And so that's also the thing. This knowledge isn't just stuffy, dusty old things you got to cram in your brain because someone's making you memorize them. These are the tools that you need to change the world.
I think Mrs. Martin is really glad you said that. We had another question in the Q&A. Someone was asking, due to the ambiguity of amino acids, can we use any base sequences that codes for a certain amino acid to build a protein?
I'm sorry, say one more time.
Someone was asking, due to the ambiguity of amino acids, can we use any base sequence that codes for a certain amino acid to build a protein?
Base sequence. So let's see. So the DNA encodes for proteins, and then the DNA, you have machinery inside of your cells that read these DNA blueprints and then convert that into a message that then gets used to build the protein directly. And so the order of the amino acids in any one protein nanobot is very precise. You need this exact string of amino acids where each bead on the string has a very carefully controlled identity. If there are any changes from the blueprint, that's what we call a mutation, and that can break the protein. Sometimes it just breaks it so it doesn't work, and sometimes it breaks it in a way that changes the function and makes it do things that are bad. Sometimes it breaks the function and changes it to make it do things that are exciting and new. That's how evolution works.
So, for students who already have that passion for whatever field they want to go into, just like you did when you were in high school, and also who are interested in, getting some of the practical experience that you've been talking about, they were wondering if you would suggest internships, and if so, how they might want to go about it.
Yeah, totally. The big advice is the thing you're excited about, start doing it as soon as you can. There are certainly many internship opportunities if you go looking for them. So there are fewer internship opportunities for high school students to get into a lab, but they definitely exist. I can do a little bit of research and send some links to you afterwards for internship programs. But another thing you could do is just go to the websites for universities, look at the research departments, and find someone in their research. Each lab will have a page that describes the research that they perform, the types of things that they're excited about, and what they work on. Read those lab websites, find something that looks interesting, and email the professor and ask if you can do an internship. They might say no, but if you email enough professors, you'll find someone who says yes. So that's maybe the big advice is people who are successful are gritty and are willing to keep trying. And so don't be afraid to keep trying. Reach out to people. Most scientists are really nice. Everyone gets where they are because people help them get there. And in particular, scientists are very appreciative of the help that they got along the way, and they're very generous in helping others. And so if you want to do science, just start emailing scientists and ask to work with them.
And, would you say that now is a good time to do that, or do you have any suggested times when students can do that?
Yeah, I mean, when you know enough about what they work on to get excited about it, that's the time.
Got it. Some people were also wondering, whether nanorobots will be designed in the future to go in the body and inject proteins to neutralize diseases.
So, yeah, there's how we get access to the medicines is really interesting and exciting. We're really just starting to enter the age of applied genetic engineering, right? The first genetically engineered humans are alive today in China. Their DNA was edited with CRISPR, and they're immune to HIV. Our bodies are also hosts to a giant community of bacteria and microbes. In fact, if you count the number of cells in your body, there are more bacterial cells than human cells. So these communities are very diverse and important for your health. They help you to digest food, they help to keep you safe. And we're just starting to understand how important they are. And the cool thing is these bacteria are much easier to genetically engineer than we are. And so you have the opportunity to genetically engineer the bacteria that live in us and on us in order to make us healthier.
Got it. And where do you sort of see the future of this gene editing going, not just with these bacteria, but also, on humans themselves?
Yeah, that's a really important question that I think many people don't know that human genetic engineering exists now. And there are many very important ethical questions that need to be asked and need to be answered. The genetically engineered humans in China, the parents of that may actually not have fully known the risks that was involved, that that their children are now exposed to, which is really quite unfortunate. And it was also done in secret because the scientists who did it knew that it would not be allowed. And I think this really highlights the importance to have these conversations openly and early, because it's happening. And if we don't have the conversations about how to ethically genetically engineer humans now, then it will be done unethically and in the dark. So these conversations are important to have.
Got it. And since it's 4 p.m., we'll be wrapping up our event right now. So thank you so much for coming, Dr. Bahl. It was really enlightening to not only learn, how you guys do protein design in your lab, but the implications of that, and also any advice you have for students who are interested in pursuing a route in science. We really appreciate it.
Had a lot of fun. Thanks everybody for the really excellent questions. And thanks again for inviting me to be with you all today, this afternoon.
Naman, would you like to share, if people enjoyed this event, how they can get involved in future ones?
Yeah, sure. So we have a Facebook page, which you guys can check out for our future events. And we also have a website, also for future event information and information about our team. So I'll be sending those in the chat. And in addition to that, we would really appreciate what you thought of this event and your other feedback. And you can feel free to fill out the feedback form, which I'll also be sending in the chat. Other than that, thank you all for coming. And we hope to see you in our next event.
Yeah, and thank you so much for coming to speak at our school, Dr. Bahl. We really appreciate it.
Had a great time. Thanks, everybody.